Okay, so we are in the week of February 1 through 3. So we are covering cells and tissues. So you'll actually have an exam this week on chapters 1, 2, and 3. Okay, so that'll be human body and orientation, basic chemistry, and cells and tissue. Okay, so that's what the first exam will be over. All right, it'll be a multiple choice exam. All right, so let's go on. All right. So I'm going to use the outline this time because it's 25 pages as opposed to 211 slides. And so I think this will be a little easier. There'll be some visual things that we can go through. But as far as the lecture exam, you mainly need to know the concepts and then the pictures. A lot of those pictures you've already seen in lab, but we'll supplement as necessary. All right, so cells, let's start there. So they are your structural units of everything that's alive, okay? And again, this comes straight from the cells and tissues week 3 module, okay? And the human body has between 50 and 100 trillion cells at any given time. So an important thing that you'll want to know is cell theory. So in cell theory, you have 4 basic components. The first is that cells your basic structural and functional unit of your organisms that are alive. Yes? Thank you. So activity of the organism is going to depend on collective activities of the cells. Okay, so all the cells working together determines your activity in the organism. So, and according to the principle of what we call complementarity, biochemical activities in cells are going to be dictated by the relative number of the specific subcellular structures. And subcellular means structures inside the cell. Okay. So if you have a lot of something, that means that cell is going to be probably advantaged in that area. If you don't have much of it, it's not going to do much of that. And the fourth, continuity of life has a cellular basis. So continuity just means it's continuous, okay? So what's happening at the cellular level influences what happens at your organismal level. That's basically what it's saying. But those are our 4 concepts in cell theory. So now that we've covered cell theory, let's talk about chemical components of your cells. So hopefully you remember a little chemistry from last week, right? So most cells are composed of 4 elements. And they are carbon, hydrogen, oxygen, and nitrogen. Okay, remember them? Okay. And actually your cells are about 60% water. Okay, so more than half of your weight is in water. Okay. So the other proteins, carbohydrates, nucleic acids, lipids make up a lot of the rest, right? All right, so let's talk a little bit about cells in terms of their anatomy. So cells typically have 3 main parts. So if we start from the inside and go out, we start with the nucleus. Then you're gonna have some cytoplasm and a bunch of stuff in it. Then you have a plasma membrane or cell membrane, okay, surrounding the cell. So for the nucleus, it is your cell's control center, okay? It's where all the direction is going on, all the management, okay, big decisions. And it has your genetic material that we call DNA, or deoxyribonucleic acid. Remember, that's the nucleic acid, right, from last week. So why is DNA important? Well, it's needed to make proteins, okay? It carries the instructions to make proteins. And DNA, let's see. All right, so here's where we are. DNA carries the instructions to make proteins. And DNA is going to be necessary for reproduction of your cells, okay? Now, in the nucleus, it itself has three regions. It has a nuclear membrane or nuclear envelope. It surrounds it. And inside, it has a nucleolus, and it has chromatin. Okay, and we'll get into what those are. But those are kind of your three main regions of the nucleus. All right. So, start with the nuclear membrane, or nuclear envelope. It's a double membrane, which means it has how many layers? Two. Okay, and surrounds the nucleus. So it is like your phospholipid bilayer in that they both have two layers. Okay. So the nuclear membrane is going to contain nuclear pores. And what are pores? Openings, right? You think about the pores in your skin, right? So they allow for exchange of material with the rest of your cell. Okay? So things can get in and out through the pores. And encloses a fluid. It's kind of jelly-like, and we call that nucleoplasm because it's inside the nucleus, just like the cytoplasm is inside the cell. Now, the second region in the nucleus we said was the nucleolus. Plural is nucleoli. The nucleus is usually going to have one or more of these nucleoli. And what happens there? Well, that's where ribosomes are assembled. Okay, it's where they're made or put together. And the ribosomes then are going to migrate to the cytoplasm. How do they get there? They go through those nuclear pores we just talked about, right? And they become sites for protein synthesis. Okay, so proteins get made at the ribosomes. Okay, third component, chromatin. So the DNA is going to be composed, or the chromatin rather, is made up of DNA and protein. Okay, that's chromatin. Take DNA, add some protein to it. And when do you see chromatin? Well, you see it when the cell is not going through division. Okay, no mitosis. When it's in mitosis, you see something else. And so the chromatin is then going to condense to form these structures called chromosomes. They're rod-like. And that's what you see when the cell divides. Okay, so when the cell's not dividing, you can see chromatin in the nucleus. When the cell is dividing, you see chromosomes. Okay? All right. All right, so let's pause and talk to your battle buddy or battle buddies and talk to them about the portions or what you've learned about characteristics of cells and then characteristics of the nucleus. Okay, so maybe one battle buddy can talk about the cell itself and the other can talk about the nucleus. And you can do this at home as well. Okay, so now that we talked about the nucleus, let's talk about the plasma membrane. Okay, plasma membrane is transparent because you can see through it, and it is a barrier for what's in the cell. And it's going to contain the components or contents of the cell. And it's going to separate those contents from the environment around it, right? It separates what's inside the cell from what's outside the cell. And I believe we talked about this briefly when we talked about phospholipids and lipids in the last chapter, but there's a concept called the fluid mosaic model. Okay, so the plasma membrane is constructed of phospholipids, cholesterol, remember those are lipids, and we have proteins, and we have carbohydrates or sugars. Okay, but the fluid part means there's movement. Okay, the membrane can move. Then you have the mosaic part, meaning that composition is variable. Okay, it's not rigid. Okay, what it has to look like. It can, different parts can have different components of those that are listed there. Okay. All right. Another part of that fluid mosaic model has to do with the phospholipid arrangement, how the phospholipids are arranged. Okay, so hopefully you remember a little bit about the structure of the phospholipids. You know, they have hydrophilic heads that are polar, they like water, and they're going to be positioned to face the inner and outer surfaces of that cell membrane or plasma membrane. And then you've got those water-hating hydrophobic tails that are nonpolar, and they're in the center or the inside of the membrane, okay, because they don't like water. Okay. So the inside of that membrane, because it is hydrophobic, makes that plasma membrane impermeable to most substances that are soluble in water. Okay? So remember, impermeable means you can't get through it, right? So if things that like water typically can't go through the middle of the membrane on their own. Okay. Now, the proteins in that membrane have different functions. Some of them are enzymes that catalyze chemical reactions. Some of them are receptors. And some of them are transport proteins. Some are channels and some are carriers. The channels are basically an opening. Things just flow through them. Carriers will pick things up and move them to the other side of the membrane. Okay, so that's what proteins do in the plasma membrane. All right, so we also have sugars or carbohydrates in the plasma membrane. So we've got glycoproteins. These are proteins with a little sugar on them. And so these are sugars that are branched and they're attached to proteins on that extracellular surface. They're on the outside of the cell membrane. Okay, you also have what we call a glycocalyx. And it's also on the surface of the cell. It's fuzzy and sticky and lots of sugar on it. Okay. So another component of the plasma membrane are that it has junctions. And what is a junction? You might know what a junction is. It's a space, but it's also a connection point. Okay, so these are ways that cells are connected to each other. So you have glycoproteins in the glycocalyx, and they serve as a glue. They're an adhesive. And you have what we call wavy contours in the membranes of cells that are next to each other, that are adjacent. And then they fit together, kind of like a tongue and groove. So it's kind of like, let's just say you have waves, and the waves in one cell match the waves in the other, so they just attach to each other, right? They interlock like fingers, so it's like that. So all right. Another way of thinking about it is kind of like Lego pieces, right? Because you have the little grooves in the Legos and you can stick them together and they just fit. Just like that. All right. Special membrane junctions get formed and they can vary depending on what their role is in the cell. Like what's their purpose, right? Remember, structure determines function. All right. So let's talk a little more about membrane junctions. Okay. So first we'll talk about tight junctions. So tight junctions, as the name suggests, it's a tight connection, which means that those junctions are impermeable. Okay, things can't get through. So why would you want that? Okay, so here you're binding cells together or connecting them into essentially leak-proof sheets. So these are cases where you don't want any leaking. And it keeps substances from passing through the space outside the cell, going between the cells. So here's where you don't want things to get in and out. Okay, so that's where you have that, you're going to have tight junctions. The next type of junction is the desmosome. And these are anchors. And essentially their role is like what does an anchor do for a boat? Hold it in the position, right? Hold it steady. So they keep the cells from getting pulled away, okay, as a result of any stress. All right, so some of these, there can be cells where there's pressure against one side of the cell. But if they have an anchor, even when that pressure comes, they're not going to get pulled away. All right, and these are created by button-like thickenings in the adjacent plasma membranes. Okay, so the desmosomes are anchors. Okay, that's the big thing I want you to think about. Okay, third type of junction are the gap junctions. All right, so they're not tight junctions, which means they're not impermeable, right? So these are junctions that are allowing for communication between cells. And they have these proteins called connexons that are essentially little hollow cylinders, like basically kind of like little straws in between the cells. And they go basically the width of the two membranes. That's how long they are. And molecules go directly through those proteins, those tubes, and go from one cell to the next. Okay? So remember, tight junctions, impermeable, no communication. Desmosomes are anchors, and gap junctions allow communication. All right? All right, so then now we have the cytoplasm. So before we get into the cytoplasm, let's take a break, and I want you to tell your partner, your battle buddy, 3 things you learned about the plasma membrane and about membrane junctions. Okay. All right, so let's do that, and you can do that at home as well. Okay, so now that we've discussed the plasma membrane and the nucleus, so the third component of the cells, let's talk about the cytoplasm. So material outside the nucleus and inside the plasma membrane is your cytoplasm. Everything between that nuclear membrane and the plasma membrane. It's where most of the things in your cell happen. three major elements of the cytoplasm. You have the cytosol, the organelles, and the inclusions. So the cytosol is the fluid in the cytoplasm. Okay, it's what's going around and suspending the other components. The organelles are like little organs performing functions inside the cell. They are your metabolic machinery. They're the ones doing chemical reactions. That's what metabolic means. And you've got inclusions. And these are just different chemical substances. That's really what that means. And these include things like nutrients that have been stored in your cells, as well as products of the cells. All right, so not much to say on the cytosol other than what's already been said, that it's a fluid, right? So let's talk about the organelles. So the organelles basically are specialized regions inside the cell that have specific functions, okay? So many of them are membrane bound, meaning they have a membrane that encloses them, like the nucleus or the cell membrane. And this compartmentalization, divvying out responsibilities to the different organelles, is critical for that organelle's ability to perform the different functions, okay? So in this case, you wouldn't want one organelle that did everything. You really want them to be specialized, okay? It's kind of like, sports, you have, on a team, you have different people who play different positions, right? So, yeah, I mean, having people who can play multiple positions is great, but at the same time, you need people who specialize in specific tasks. So the first organelle that we are going to cover is the mitochondria, or are the mitochondria. So mitochondrion is singular, mitochondria is plural. So the mitochondria are the powerhouses of the cell. They're the engine, essentially. Well, probably furnace is a better description. They're generating the energy. All right. So they're changing shape as they do this. In the wall of the mitochondrion is a double membrane, just like it is in the nuclear membrane and the cell membrane. But this membrane has what we call cristae on the inner membrane. All right. And it creates all these ridges on the inside of the mitochondria. Mitochondria can carry out reactions where oxygen gets used to break down food. And those reactions lead to production of ATP, adenosine triphosphate, to create energy for your cells. Okay, so mitochondria, powerhouses of the cell, create ATP for energy. Okay? And we have ribosomes. Ribosomes have two lobes to them. There are two subunits. And they're made up of protein and ribosomal RNA. And so they've got protein and nucleic acid. And they are sites where protein is made, sites of protein synthesis. Where do you find ribosomes? Well, I'm glad you asked. You can find them in two locations inside your cells. First location would be free floating on their own in the cytoplasm. Okay, the second location where you find ribosomes would be attached to the rough endoplasmic reticulum. Okay, and that's what makes it rough. As we'll learn later with the endoplasmic reticulum, if it doesn't have ribosomes, it's considered smooth. Okay. All right, and that is a good segue to our next organelle, the endoplasmic reticulum. So the rough endoplasmic reticulum has ribosomes on it, And because it has ribosomes, its role is to make protein. Also, there are transport vesicles, and we'll learn more about those, but they're basically little spheres or bubbles. They're not really bubbles, they're spheres, but they look like a bubble. And they move proteins to other locations inside the cell. And rough ER is going to be abundant, means you're going to have a lot of rough ER in cells that their role is to make and export proteins. Remember, what does exporting mean? Do you already know from economics or social studies? Send out somewhere else, right? So that's what the rough endoplasmic reticulum is for. The smooth endoplasmic reticulum, so its function is to metabolize lipids. So it's chemical reactions involving lipids. Okay, that's what the smooth endoplasmic reticulum does. Why? Because it doesn't have ribosomes. It also detoxifies drugs and pesticides, makes them less damaging for your body. All right. Next, we have the Golgi apparatus. And it's going to appear as a stack, essentially, of flattened membranes that are associated with tiny vesicles. Okay? And they're going to modify and package proteins that arrive from the rough ER. How do they get there? Through those transport vesicles we just talked about, those spheres. And they're going to produce different kinds of packages. Okay, so they can produce secretory vesicles, and that just means that they have secretions, which means they release them outside the cell. Okay, second pathway would be in-house proteins and lipids, and in-house in this context means they don't get sent out to another cell; they stay in the cell. And then third pathway is going toward or making lysosomes. And we'll learn more about lysosomes now, but those are basically the three different pathways that packages can take from the Golgi apparatus. Okay, they can be sent out of the cell, the products can be used inside the cell, or they can be sent to the lysosomes. And lysosomes are membranous bags, essentially, that are packaged by the Golgi apparatus. They have enzymes in them that are produced by ribosomes. Remember, what do ribosomes make? Anybody remember? Protein. So enzymes are proteins, right? So enzymes catalyze chemical reactions. And what do they do? In the lysosomes, or the lysosome enzymes are going to digest worn out or cell structures that you can't use anymore. Okay? And they also are going to house phagocytes, which are cells that eat things. Phago means eat. So they're cells that eat things to dispose of things like bacteria and other cellular trash or debris. Okay? So then we have the peroxisomes. So the peroxisomes these are membranous sacs, and they have a different kind of enzyme. They have oxidases. Okay, remember, anytime you see ASE, that means protein. And their role is to detoxify things that are harmful to you, things like alcohol as well as formaldehyde. Okay? And they're going to break down things called free radicals. And these are just basically chemicals that are highly reactive. Okay? You don't need to know much about them other than that. And these free radicals get converted to hydrogen peroxide, hence peroxisomes. and then their hydrogen peroxide gets converted to water. Okay, so you take something that's dangerous and convert it to something that's not dangerous. Okay? And the Proxy Zones are going to replicate or copy themselves by pinching in half. Or they can butt off of the endoplasmic reticulum. Okay? So we also have the cytoskeleton. Cyto means cell, skeleton's a structure. So it's a network of protein structures that are extending throughout your cytoplasm. They're going to provide your cell with a framework. It's an internal framework inside your cell. 3 components. The largest components are microfilaments. In between, you have intermediate filaments. And in smaller ones, you have microtubules. Okay, those are the 3 components of your cytoskeleton. All right, so then we also have centrioles. And centrioles then are going to be involved in cell division. They're rod-like bodies, and they're made up of those microtubules we talked about before with the cytoskeleton. And then they also are going to create or generate microtubules. Okay, and so what do? Well, they are going to control or direct the formation of the spindle fibers or mitotic spindle during cell division. All right, and so as we talk about cell division, we'll learn essentially the spindle fibers will attach to chromosomes and move them to opposite sides of the cell so that then you can divide the cell in half. Okay? That centrioles are involved in cell division. Okay, that's what I want you to know there. We'll also have cellular extensions on the outside of the cell, on the surface. You have the cilia, the flagella, and the microvilli. The cilia are going to move materials across the cell surface. So they just kind of blow things over the surface. Okay? And they're located in your respiratory system, help you move your mucus. Okay, that's one place where you'll find cilia. You also have flagella, and their role is to actually propel the cell to move the cell itself. Okay, the cilia are moving materials away from it or across the surface. Flagella actually move the cell itself. So the only flagellated cell in your human body or in a human body would be sperm cells. Okay, so then we have microvilli. These are small finger-like projections of the plasma membrane. What do? Well, they increase the surface area of the plasma membrane because they've added these little ridges, okay? And that helps when you need to absorb things, right? Like in your small intestines. So areas where you need to absorb things, you might have microvilli. So you can absorb, like for example, you absorb the food you eat in the small intestine. All right. So let's pause here and talk to your battle buddy, and I want you to tell your battle buddy the names and functions of 3 organelles. Okay, each one of you should be able to do that. And you do this at home as well. So now we've covered the organelles in the cell, let's talk about the diversity of cells, all right? Because they're not all the same. So human bodies can house over 200 different types of cells, okay? So just like all people are not the same, all cells are not the same. All right, so cells are going to vary in length from 1/12,000th of an inch to over 1 yard, right? So you have some nerve cells in your body that are actually 3 feet long, okay? You actually consider the whole thing one cell. And cell shape is going to reflect the specialized function, right? So function, shape, structure, all those things are related. Cells, you have certain cells, and their role is to connect different parts of your body. So the fibroblasts are going to secrete cable-like fibers, okay? And blast, anytime you see B-L-A-S-T, those are cells that typically are creating things, okay? These are cells that are building. And you also have erythrocytes. They're your red blood cells, and they're essentially carrying oxygen from one part of your body to another. So in that sense, they're connected. All right, and these are both examples of cells that actually will form what we call connective tissue. All right, we also have cells that line and cover organs in your body. So examples of those are epithelial cells. Okay, and they come together as a sheet or a layer. You have intermediate fibers, and they can resist rubbing or tearing when they're rubbed or pulled. All right, and that's epithelial cells come together to form epithelial tissue. You also have cells that move organs as well as other parts of your body. Okay, so example here, you got skeletal muscle, which is part, these are different types of muscle tissue, smooth muscle, smooth muscle cells form smooth muscle tissue. And these have contractile components, and they can allow the cells to shorten with some force. You also have cells that are involved in storing nutrients. So example there, your fat cells are adipocytes. Okay, and so these essentially have lipid droplets in them, and they're stored inside the cytoplasm of the cell. All right, you also have cells that help you protect yourself against foreign invaders, right? Help you fight disease, like your macrophages. And these are phagocytes, cells that eat things, and they're going to digest or break down infectious microorganisms. Okay. You also have cells that gather information and control different functions in your body. All right, and so example of that are your neurons, or nerve cells. So they can receive information or transmit messages to other parts of the body. Okay. All right. We also have reproductive cells. And these reproductive cells include things like oocytes in females. These are cells, the largest cells that typically you'll see in the body. And they can divide and become an embryo if they get fertilized. Males have sperm cells, and they're built for swimming to the egg to fertilize it. And how they do that? Through the flagella that we learned about before. And it acts essentially as a whip that kind of pushes the sperm cell forward. It gives it what we call motility or movement. All right, so now we discussed cell physiology. Let's talk a little bit about, or cell diversity. Let's talk about cell physiology. All right. So some of the different things that cells can do, they can metabolize things. They can basically metabolize, has to do with chemical reactions. They can digest or break down food. They can get rid of or dispose of waste. They can reproduce, right? They can divide. They can grow, get larger in size, and they can move, or some cells move, and they can respond to stimuli. Okay, so these are all different kinds of things cells can do. All right, so now that we've kind of covered cells pretty strongly, let's also kind of talk a little more specifically about the membrane and specifically how substances are transported, okay, across the membrane. So in order to understand that, you have to understand what solutions are. So solutions are homogeneous mixtures, okay? And those mixtures can have two substances or more, or two components or more. And so you have one that's the solvent, that's what things dissolve in. In most cases in your body, it's going to be water. And you have solutes, and these are the smaller quantities in the solution that dissolve in the solvent. All right, so if we have salt water, salt is the solute, water is the solvent, right? Because salt's dissolving in the water. So the intracellular fluid is fluid inside the cell. And that includes your cytosol and your nucleoplasm, okay, because those are all fluids inside the cell. And in that solution, you're going to have things like gases, right, like oxygen, carbon dioxide. You have different nutrients, right, food that's been broken down. You have salts that get dissolved in water, right, different ions. Now, outside the cell, you also have fluid. We call that interstitial fluid. There you've got, again, nutrients. You may also have hormones, right, that can tell your cells to do certain things. Neurotransmitters, which, again, kind of have a nervous, they can, stimulate nerves to do certain things. All right, we'll learn more about that when we get to the nervous system. Also salts or ions and waste products. Okay, you'll find all of those things in the interstitial fluid outside the cell. Okay, important quality, I want to make sure you highlight, underline, whatever you do, but make a note that the plasma membrane is selectively permeable. Okay, it's important that you understand that concept. It is selectively permeable. So what does selective mean?
It can or can't.
Right. So like when you say someone has selective hearing, it's like they hear what they want to hear, right? So it's kind of like that with the plasma membrane as well. So some materials can go through the membrane, others cannot. For example, you can get nutrients that go into the cell. Things that you don't want to go in sometimes are kept out. So how can we transport things? Well, there's two basic ways that we transport things. All right. So let's see. Yep. So it can be passive or active. If it's passive, you don't need any ATP. No energy is needed to do this. Okay, it's going to happen naturally. If it does require ATP or energy, it's active. Okay, that's active transport. So we'll start with the passive processes. Yeah. Okay. So particles are going to move themselves evenly in the distribution in a solution. All right, we call that diffusion. Okay, and so the driving force here, what's making that happen is kinetic energy, which again, kinetic has to do with motion, right? Like kinesiology has to do with study of people moving. All right, and it's causing molecules to move around randomly. So diffusion then is molecules moving from areas of high concentration to areas of low concentration. All right, so it's also moving down the concentration gradient. So, important for you to understand how molecules move okay, relative to their size and their temperature. So you have small molecules can move faster, right? Okay, it's like if you're going to throw, like which one would you be able to throw further, a baseball or bowling ball? The baseball, right? It's going to be easier, right? And if you had to hold a ball and move with it, which one is you're going to move faster with, the light bowling ball or the big heavy bowling ball? The light one, right? Also, the warmer the molecule, the faster your diffusion is going to happen. So, because heat is energy, right? Heat. And so when you have more heat, okay, the things move faster or slower? Faster, right? So if I want to get up and run, I'll run even when it's cold, and I'm probably going to run a little bit faster, more quickly. You know, my muscles kind of get moving a little bit better when it's warmer, right? All right, so more motion, more heat, more energy, more motion. It's faster. So, all right, here's an example of diffusion. All right, it's 1 o'clock in the afternoon, right? You're probably a little sleepy, a little tired. All right, you get that cup of coffee for the afternoon, put some sugar in it. Okay, but you're not going to stir it, right? You're just going to leave that sugar in there and let it sit. Okay, leave it for long enough, that coffee is going to taste sweet. Okay, because the molecules moved by diffusion. It sweetened up your cup. Molecules are going to move through diffusion under certain criteria, certain things, if certain conditions are in place. Okay, so if the molecules are small enough to go through the pores in the membrane, you'll get diffusion. If the molecules are soluble in lipids, okay, if they can go through the membrane, right, they will diffuse. And if they can be assisted by some carrier protein in the membrane, they'll diffuse. So they can either go through a channel, they can go straight through the membrane, or they can go through a carrier protein that picks them up and takes them across. Okay? So if any of those conditions are true, you'll get diffusion. If not, then you don't. So there's different kinds of diffusion. So you have simple diffusion. Okay, no assistance is needed here. If the solutes are soluble in lipids, which means they can go through the lipid membrane, are small enough to go through pores in the membrane, you get simple diffusion. Okay, so then there is osmosis. And osmosis means you have simple diffusion of water. So anytime you see water involved, if water is the one what's diffusing, it's osmosis. So osmosis is the diffusion of water. Osmosis is the diffusion of water. Osmosis is the diffusion of water. Thank you. All right. So this should say highly polar water molecules are going easily across the plasma membrane through proteins called aquaporins. Okay, they're just little pores or little channel proteins. Protein in the water itself is going down its concentration gradient. So wherever the concentration of water is higher, it's going to move to where the concentration of water is lower. Okay, so then we got what we call isotonic, hypertonic, and hypertonic solutions. Okay? So iso means same. So the solutions are gonna have the same solute in water concentrations as the cell does. So what's outside the cell is the same concentrations inside the cell. So you don't see any visible changes because the concentrations are equal. Then you have hypertonic solutions, and hyper means more, right? Like hyperactive is more active, or higher concentration. So the hypertonic solutions have more solutes than the cells do. All right, so the cells shrink because if there's more solute in the solution, it means there's less water. So water is going to go to where it's high to where it's low. So it goes out, cell shrinks. Hypertonic solutions have fewer solute molecules, which means they have more water than the cells do. So the cells then are going to expand, right? And if they expand too much, they'll pop open, they'll bust open. All right, another type of diffusion, we've got facilitated diffusion. All right, so facilitated diffusion, we're transporting lipid insoluble in large substances. All right, and the glucose gets transported via facilitated diffusion. There's a facilitator involved. These protein membrane channels or protein molecules act as carriers, pick up the molecules and move them across the membrane. All right. And we've got filtration. So with filtration, water molecules and solutes actually get forced across by pressure. We call that hydrostatic pressure. So it's fluid pressure. But you have to have a pressure gradient, which means the pressure is higher on one side than the other. Filtration actually happens in your body. Okay, and it happens with your kidneys. Okay, your kidneys actually filter waste out. Okay, because the pressure is higher. All right, the pressure is higher essentially in your blood vessels than it is in your urinary system. Okay, so it's pushing materials out that you don't need. All right. So quickly, I want to discuss active processes. So active processes, the big thing I want you to see is, again, if the substances are too big to go through the membrane, they can't dissolve, they can't go through the lipid bilayer. All right, they don't like lipids. Yeah, they may have to go against the concentration gradient and go to an area where the concentration is actually higher. You're gonna need active transport. It's gonna require ATP to do that. All right. So big thing here, example of active transport is the sodium potassium pump. In a sodium potassium pump, you're pumping sodium out and bringing potassium in, and it takes ATP to do this. Okay, because you're going against their concentration gradients. You're actually moving toward higher concentration. All right. So we will talk about vesicular transport next time. All right, but go ahead and do your exit tickets, and we will pick up the vesicular transport next time.